Hello, welcome to Biology, the scientific study of life. Humoral and cell-mediated immunity defend against different types of threats. Early evidence that substances in the blood, plasma, and lymph play a role in acquired immunity came from experiments performed near the end of the 19th century. Researchers transferred such fluids long ago called humors from an animal that had recovered from an infection by a particular microbe to another animal that had not been exposed to that microbe. Later, when the second animal was infected with the same microbe, it did not become ill. The investigators had transferred what we now know were secreted antibodies from one animal to the other. They also found that immunity to some infections could be passed along only if certain cells, later identified as cytotoxic T cells, were transferred. These and many other studies led to the current understanding that acquired immunity includes two branches. The humoral immune response involves the activation and colonial selection of B cells, resulting in production of secreted antibodies that circulate in the blood and lymph. The cell-mediated immune response involves the activation and colonial selection of cytotoxic T cells, which directly destroy certain target cells. Notice this figure. Helper T cells and B cells and secreted cytokines activate. Outlines the roles of the major participants in the acquired immune responses. Central to this network of cellular interactions is the helper T cells, as shown here, which responds to peptide antigens, displayed an antigen on antigen presenting cells, and in turn stimulates the activation of nearby B cells and cytotoxic T cells. Helper T cells responds to nearly all antigens. When a helper T cell encounters and recognizes a class or two class or second class MHC molecule antigen complex on an antigen presenting cell, the helper T cell proliferates and differentiates into a colon of activated helper T cells and memory helper T cells. A surface proteins called CD4 present on most helper T cells binds the class 2 or the second class MHC molecule. These interactions helps keep the helper T cell and the antigen presenting cell joined while activation of the helper T cell proceeds. Activated helper T cells secrete several different cytokines that stimulates other lymphocytes, thereby promoting cell-mediated and humoral responses. The helper T cell itself is also subject to regulation by cytokines. For instance, as a dendritic cell presents an antigen to a helper T cell, the dendritic cell is stimulated to secrete cytokines that, along with an antigen, stimulates the helper T cell to produce its own set of cytokines. The class or second class MHC molecules recognized by helper T cells are found mainly on dendritic cells, macrophages, and B cells. Dendritic cells are particularly effective in presenting antigens to naive helper T cells, so called because they have not previously detected an antigen. In other words, dendritic cells are important in triggering a primary immune response. Dendritic cells are located in the epidermis and in many other tissues where they efficiently capture antigens. They then migrate from the site of infection to various lymphoid tissues where they present antigens via second class or class 2 MHC molecules to helper T cells. Macrophages play a key role in initiating a secondary immune response by presenting antigens to memory helper T cells, while B cells primarily present antigens to helper T cells in the course of the humoral response. Notice also this is a figure that shows that the central role of the helper T cells in humoral and cell-mediated. Closer look and dendritic cell bacterium. Notice that peptide antigen cell, second cell MHC molecule. Notice that CD4 here, and notice helper T cells. After notice picture or after a dendritic cell engulfs and degrades a bacterium, it displays bacterial antigen fragments, peptides, complexed with a class or second class MHC molecule on the cell surface. A specific helper T cell binds to the displayed complex via its TCR with the aid of CD4. This interaction promotes secretion of cytokines by the dendritic cell. Notice that. And notice too proliferation of the T cell stimulated by cytokines from both the dendritic cell and the T cell itself gives rise to a colon of activated helper T cells, not shown here, all with receptors for the same MHC antigen complex. Notice 3. The cells of this colon secrete other cytokinases that help activate B cells and cytotoxic T cells. Notice this represents cell-mediated immunity attack on infected cells, and this one humoral immunity secretion of antibodies by plasma cells. Cytotoxic cells response to infected cells and cancer cells. Cytotoxic T cells, the effectors of cell-mediated immunity, eliminate body cells affected by viruses or other intercellular pathogens, as well as cancer cells and transplanted cells, fragments of non-self proteins synthesized in such target cells associate with the class 1 or first class MHC molecules and are displayed on the cell surface, where they can be recognized by cytotoxic T cells. A surface protein called CD8 present on most cytotoxic T cells greatly enhances the interactions between a target cell and a cytotoxic T cell binding of CD8 to the side of a class 1 MHC molecule helps keep the two cells in contact during activation of the cytotoxic T cell. Thus, the role of a class 1 MHC molecules and CD8 is similar to that of a second class MHC molecules and CD4, except that different cell types are involved. When a cytotoxic T cell is selected by binding to class 1 MHC molecule antigen complexes on an infected body cell, the toxic T cell is activated and differentiates into an active killer. Cytokines secreted from nearby helper T cells promote this activation. The activated cytotoxic T cell then secretes proteins that act on the bound infected cell, leading to its destruction. Notes as shown in this figure. The death of the infected cell not only deprives the pathogen of a place to reproduce, but also exposes it to circulating antibodies, which mark it for disposal. After destroying an infected cell, a cytotoxic T cell may move on and kill other cells infected with the same pathogen. In the same way, cytotoxic T cells defend against malignant tumors Because tumor cells carry distinctive molecules, tumor antigens not found on normal body cells, they are identified as foreign by the immune system. Class 1 MHC molecules on a tumor cell display fragments on tumor antigens to cytotoxic T cells. Interestingly, certain cancers and viruses such as Epstein-Barr virus actively reduce the number of class 1 MHC molecules on affected cells, helping them escape detection by cytotoxic T cells, but the body has a backup defense. Recall that natural killer innate cells, part of the body's non-specific innate defenses, can induce apoptosis in virus infected and cancer cells. B cells are response to extracellular pathogens. Antigens that elicit a humoral immune response are typically proteins and polysaccharides present on the surface of bacteria or incompatible transplanted tissue or transfused blood cells. In addition, for some people, proteins or foreign substances such as pollen or bee venom act as antigens that induce an allergic or hypersensitivity humoral response, as shown in this figure. Notice that depicts have events in the humoral response to a typical protein antigen. The activation of B cells is aided by cytokinesis secreted from helper T cells activated by the same antigen stimulated by both an antigen and cytokinesis. The B cells proliferates and differentiates into a colon of antibody secreting plasma cells and a colon of memory B cells. When an antigen first binds to receptors on the surface of a B cell, the cell takes in a few of the foreign molecules by receptor mediated into cytosis in a process similar to antigen presentation by macrophages and dendritic cells. The B cell then presents antigen fragments to a helper T cell. This achieves the direct cell-cell contact critical to B cell activation. Notice the steps here. Note, however, that a macrophage or dendritic cell can present peptide fragments from a wide variety of antigen, whereas a B cell internalizes and presents only the antigen to which it specifically binds. Antigens that induce antibody production only with assistance from helper T cells, as shown in this figure. Notice that the 5 classes of immunoglobulins. Antigens that induce antibody production only are here known as T-dependent antigens. Some antigens, however, can evoke B cell responses without involvement of helper T cells, such as in unindependent antigens include the polysaccharides of many bacterial capsules and the proteins that make up bacterial flagella. Apparently, the repeated subunits of these molecules bind stimulously to several antigen receptors on a single B cell, providing enough stimulus to activate the cell without the helper of cytokinesis. The response to T-independent antigens is very important in defending against many bacteria. However, this response is generally weaker than the response to T-dependent antigens and generate no memory B cells. Most antigens recognized by B cells contain multiple epitopes. For this reason, exposure to a single antigen's norm stimulates a variety of different B cells, each giving rise to a colon of thousands of plasma cells. Or the plasma cells in one colon secrete antibodies specific for the epitopes that provoke their production. Each plasma cell secretes an estimated 2,000 antibody molecules per second over the cell's 4 to 5 day lifespan. Antibody classes. A secreted antibody has the same generally Y-shaped structures as a B-cell receptor, but lacks a transmembrane region that would anchor it to the plasma membrane, although the antigen binding sites on an antibody are responsible for its ability to identify a specific antigen. The tail of the U-shaped molecule formed by the constant C regions of the heavy chains is responsible for the antibody's distribution in the body and for the mechanisms by which it mediates antigen disposal. There are 5 major types of heavy chain constant regions, and these determine 5 major classes of antibodies. The class names are based on the alternate term for antibody immunoglobulin Ig. The structures and functions of these antibodies classes are summarized in this figure. Note that 2 classes exist primarily as polymers of the basic antibody molecule, IgM as a pentamer and IgA as a dimer. The other 3 classes, IgG, IgE, and IgD, exist exclusively as monomers. Notice that. Produced in the body following exposure to a microbial antigen are polyclonal. In contrast, other antibody tools are monocolonal. They are prepared from a single colon of B cells growing in culture. All the monoclonal antibodies produced by such a culture are identical and specific for the same epitope. On an monoclonal antibodies are particularly useful for tagging specific molecules. For example, certain types of cancer are treated with tumor-specific monoclonal antibodies bound to toxin molecules. The toxin-linked antibodies carry out a precise search and destroy mission, selectively attaching to and killer tumor cells. Antibody-mediated disposal of antigens. The binding of antibodies to antigens is also the basis of several antigen disposal mechanisms. In the as shown in this figure, antibody-mediated mechanisms of antigen disposal. In the simplest of these viral neutralization antibodies bind to certain proteins on the surface of a virus, thereby blocking the virus's ability to infect a host cell. Similarly, antibodies may bind to a pathogenic bacterium, coating much of the bacterial surface in a process called opsonization. The bound antibodies enhance macrophage attachment to the microbes and thus increase phagocytosis. Antibody-mediated agglutination, clumping of bacteria or viruses from aggregates that can be readily phagocytosed by macrophages. Agglutination is possible because each antibody molecule has at least 2 antigens binding sites that can bind to identical epitopes on separate bacterial cells or virus particles, linking them together because of their pentameric or because of pentameric structure. IgM antibodies can link together 5 or more viruses or bacteria, as shown here. Notice that, closer look. Binding of antibodies to antigen inactivates antigens by, notice that, enhances phagocytosis and cell lysis leads to. In the similar process of precipitation, antibodies cross-link soluble antigen molecules dissolved in body fluid, forming immobile aggregates that are disposed of by phagocytes. And as notice also substances on many microbes activate the complement system as part of the body's innate defenses. The complement system or the complement system also participates in the antibody-mediated disposal of microbes and transplanted body cells, in this case binding of antigen-antibody complexes on a microbe or foreign cell to one of the complement proteins triggers a cascade in which each component activates the next. Ultimately, activated complement proteins generate a membrane attack complex MAC that forms a pore in the membrane. Ions and water rush into the cell, causing it to swell and lyse. Whether activated as part of innate or acquired defenses, the complement cascade results in lysis of microbes and produces activated complement proteins that also promote inflammation or stimulate phagocytosis. Antibodies promote phagocytosis in several ways. Recall that phagocytosis enables macrophages and dendritic cells to present antigens and stimulate helper T cells, which in turn stimulate the very B cells whose antibodies contribute to phagocytosis. This positive feedback between the innate and acquired immune systems contributes to a coordinated, effective response to infection. Immunity conferred by natural exposure to an infectious agent is called active immunity because it depends on the action of person's own lymphocytes and the resulting memory cells specific for the invading pathogen. Active immunity also can develop following immunization, after called vaccination from the Latin vasca, cow. The first vaccine consisted of the virus causing cowpox, a mild disease usually seen in cows, but also occasionally in humans. In the late 1700, back to antibody-mediated mechanisms of antigen disposal. Notice here represent this the binding. Notice the binding here of antibodies of antigen marks microbes, foreign particles, and soluble antigens for inactivation or destruction. Following activation of the complement system, the membrane attack complex MAC forms pores in foreign cells. The pores allow ions and water to rush into the cell, leading to its swelling and eventual lysis. Notice that macrophage, and notice enhances here bacteria agglutination of antigen bearing particles such as microbes, and notice precipitation of soluble antigens, and notice activation of complement system and pore formation. Pore MAC complement proteins and foreign cell leads to lysis here. Notice also viral neutralization blocks binding to hosts, and notice also opsonization increases phagocytosis. Back to the 5 classes of immunoglobulins. All classes consist of similar U-shaped molecules in which the tail region determines the distribution and functions characteristic of each class. IgM and IgA antibodies contain a G chain. unrelated to the gene segments that helps hold the monomer subunits together as gene antibodies is secreted across a mucous membrane. It requires a circulatory component that protects it from cleavage by enzymes. Notice that. Back to the humoral immune responses. Notice these are bacterium, peptide antigen, and notice also macrophage, class 2 or second class MHC molecule, and CD4, TCR, helper T cell. Notice that these are notice most protein antigens require activated helper T cells to trigger a humoral response, either a macrophage, notice that, or a dendritic cell can activate helper T cells. The TEM of a plasma cell reveals abundant endoplasmic reticulum, a common feature of cells dedicated to making proteins for secretion, TCR or TC receptor, and the positive indicates stimulation. Notice that positive here. Cytokinesis, activated helper T cell, and the colon of a memory B cell, and colon of plasma cells secreted antibody molecules. Notice also. This figure represents, notice that the killing action of cytotoxic T cells after increased interacting with a target cell, such as an infected body cell or a cancer cell. An activated cytotoxic T cell releases perforins and proteolytic enzymes, granzymes, that promote death of the target cell. The colorized CEM shows here, notice that, cancer cell with a perforin induced in the early stages of apoptosis, TCR T cell receptor, cytotoxic T cell here. Cytotoxic T cell and TCR here, and class 1 MHC molecular target cell. Notice these are the steps. Steps 1 or step 1, a specific cytotoxic T cell binds to a class 1 MHC antigen complex on a target cell via its TCR with the aid of CD8. This interaction, along with the cytokinesis from the helper T cells, leads to the activation of the cytotoxic cell. Notice too, the activated T cell releases foreign molecules which form pores in the target cell membrane, and proteolytic enzymes, granzymes, which enter the target cell by the endocytosis. Third step is notice that the granzymes initiate apoptosis within the target cell, leading to fragmentation of the nucleus, release of small apoptotic bodies, and eventual cell death. The released cytotoxic T cell can attack other target cells. Back to the central role of helper T cells in humoral and cell-mediated immune responses. Notice also the CEM of dendritic cell reveals its long branching extensions, reminiscent of the dendrites of nerve cell. Dendritic cells are primary antigen presenting cells. During the primary response to an antigen, TCR, which equal T cell receptor, and the positive indicates stimulation here. After dendritic cells engulfs and degrades a bacterium, it displays bacteria antigen fragments, peptides, complexed with a class 2 MHC molecule on the cell surface. A specific helper T cell binds to the displayed complex via its TCR with the aid of CD4. This interaction promotes secretion of the cytokines by the dendritic cell. Notice that. Notice also 3 proliferation of the T cells stimulated by cytokines from both the dendritic cell and the T cell itself gives rise to a colony of activated helper T cell, all with receptors for the same MHC antigen complex. And the third one, the cells in this colon secrete other cytokines that help activate B cells and cytotoxic T cells. Okay, thanks for listening.